again. In this module, we'll be looking at certain experiments that were done to prove DNA as a genetic material, in which we'll be talking about the first set of experiment, which is called as Griffith's transformation experiment. So let us see what is this experiment all about. Now, experiments to prove DNA was genetic material was basically done using several bacteria and viruses. Now, in this first set of experiment, which is called as the Griffith's experiment, was conducted in the year 1928 by this guy right here, who was a British physician, and his name was Frederick Griffith. Hence, it is called as the Griffith experiment. Now, Griffith used a strain of bacteria called as Diplococcus pneumoniae. Now, it is a bacteria that is known to cause pneumonia in mammals. If you look at the bacteria under microscope, what you see are two spherical cells attached together, right? And that is why the name is Diplococcus. So it's a bacteria which has two cells attached to each other, Diplococcus, and is known to cause pneumonia in mammals. Now, Griffith basically used two different types of bacteria. Okay, the first type was the rough type. Now, it is called as the rough type because it produced rough colonies. Why were the colonies rough? Because this bacteria was non-capsulated, and hence it produced rough colonies on a medium. The other type was the smooth type, or the S type. Now, it is called as a smooth type because it produced smooth colonies on the medium. Now, why it produced smooth colonies? Because these cells were capsulated, and because of the presence of the capsule, the colonies formed on the medium were smooth. So you're looking at the rough type and the smooth type, two different types of Diplococcus pneumoniae that Frederick Griffith used for his experiment. He conducted his experiments in 4 different steps. Let us see these steps ahead. In the first step, what he basically did was he took an inoculum containing the S-type bacteria and he injected these cells into a healthy mouse. Now, when the S cells were injected in the healthy mouse, what happened was the mouse developed pneumonia. And because of pneumonia, the mouse actually died. And then Griffith concluded that the S-type bacteria is virulent, which means it causes death, or pathogenic because it causes the disease pneumonia. Okay? In the next step, what did he do? In the next step, Griffith took another inoculum which contains the R-type bacteria, which means the bacteria that forms rough colonies. He took another healthy mouse and injected the R cells into the mouse. Now, this mouse did not develop any pneumonia, and as a result of which the mouse lived or survived. So he concluded that the R-type is avirulent, which means does not kill, or non-pathogenic because it did not cause pneumonia. Correct? Moving on. In step 3, what he did was he took the S-type and he heated it. So you call this as heated killed S-type cells. Now, when the cells of the heated S-type were injected into another healthy mouse, what was seen was the mouse developed no pneumonia because the S cells, which were virulent, were now killed before injecting into mouse, and therefore no pneumonia, and the mouse survived or lived. In step 4, what he did was he took this heat-killed S-type bacteria and he mixed it with the live R-type. The combination of the two was injected in yet another healthy mouse, and was observed that the mouse developed pneumonia and was killed, or it died. Does that make sense? Now, you know that singly S-type when killed cannot cause pneumonia. Singly R-type also cannot cause pneumonia, but when the two were mixed, what really happened was pneumonia was developed in the mouse and the mouse died. Let us recollect the 4 steps once again. In the first experiment, the R cells were injected in a healthy mouse and the mouse lived because no pneumonia. In the second experiment, when the S cells were injected, the mouse died because of pneumonia. In the third set, when the S cells were heat killed, the mouse still survived, correct, because no pneumonia. And finally, in the 4th experiment, when the R cells were mixed with the heat-killed S cells, the mouse developed pneumonia and was dead. Fine. And when the blood samples were collected from this mouse in the 4th experiment, it showed colonies of live S-type. Okay? Here you should know that this change was due to a phenomena other than mutation, which Griffith called transformation. So let us see what he concluded. Griffith basically said that something got transferred from the heat-killed S strain to the R strain, which transformed the live R strain into live S strain. And Griffith called this something as transforming principle. Okay, so what is transformation? Nothing but when a host organism takes in foreign DNA and expresses the foreign gene, the phenomena is called as transformation. It was later proved that this transforming principle is DNA, but Griffith did not say that. Okay, so let us see the final conclusions of Griffith's experiment. Now, according to Griffith, or you can say Griffith discovered transformation and he first showed gene action. He also said that the R-type bacteria will basically take the transforming principle from outside and it will get converted to a particular bacteria which can develop a smooth polysaccharide coat around it, which is also called as capsule, which means the R-type now turned into the S-type and it became virulent and caused pneumonia. Now this S-type will then be transmitted to the next generation, and what you will see are many colonies of the S-type bacteria. Is it okay? Which means this transforming principle has to be the genetic material. But what was picked up by the living R-type? Was it the protein? Was it lipid? Was it carbohydrate? Was it RNA? Or was it DNA of the S-type that changed the R to the S? To prove and conclude this part, another set of experiment was done, which we'll be talking about in the next module. Okay? So in this module, we looked at certain experiments which will conclude or prove that DNA is genetic material. We first introduced Griffith's transformation experiment. Now in the next module, I'm going to talk about an experiment called as the Avery, MacLeod, and the McCarthy's experiment. Thank you and take care.